Hello my dear bachon and welcome back. I know, exams are just around the corner, but absolutely no tension. Your favorite tutor is here and together we are gonna make this chapter feel like a total piece of cake.
So let me ask you a fun question. What do you really think you're made of? Is it the roti your mom makes? The dal? Or, let's be honest, maybe it's all that Maggie you eat late at night, huh? It's a funny thought, isn't it? Well, today we're going on a little detective journey together. We're going to investigate what's really inside all living things. Yep, including us. And I promise, by the end of this super short explainer, you'll have a solid grip on this whole biomolecules chapter, and you'll feel way more confident for your exam. So, Cello, let's begin. Okay, so first things first, if we're going to be detectives, we need to know how to find the clues, right? So how do scientists actually figure out what's inside a living cell? Well, it's all about a cool process I call the Great Separation. Now, this might sound super complicated, but trust me, it's basically like cooking in a science lab. Seriously, imagine you take a vegetable and grind it in a mixer to make chutney. It's the same thing. We just add a mild acid to help break everything down. Then you strain the whole goopy mixture through a fine cloth. You know, just like how paneer is made at home. Easy peasy, right? And when you do this, you get two sets of clues. The liquid that filters through, that's called the acid-soluble pool. Soluble just means it dissolves, so this part has all the small stuff, the micromolecules. And the thick paste, the stuff left behind on the cloth, that's the acid-insoluble fraction, and it has all the big stuff, the biomacromolecules. The key thing to remember is just this. The acid dissolves the small molecules, but the big ones get left behind. Simple. Okay, so now for the exciting part. Let's meet the superstars that were hiding in that thick, insoluble paste. These are the big four macromolecules that basically run the entire show inside our cells. And first up, we have proteins. I want you to think of proteins as the busy workers of your cell. They're like the employees who do everything, seriously. From building things to fighting germs, they are the busiest superstars of all.
So what exactly is a protein? Well, it's basically a long chain, kind of like a necklace. And each little bead on this necklace is a molecule called an amino acid. Now, here's the cool part. There are 20 different types of these amino acid beads. Because it's made of different beads, we call a protein a heteropolymer. Hetero just means different. See? Simple as that.
But what's really amazing is that this long necklace doesn't just stay a straight line. No way! As you can see right here, it starts to twist and turn and fold in on itself until it makes this very specific, complex 3D shape, almost like a crumpled ball of wool. And this final, unique shape, that's the secret. That shape is what allows each protein to do its specific job perfectly. And believe me, their jobs are so important. You've got collagen, which gives our skin its structure, and hey, remember this one, it's the most abundant protein in the whole animal world. Then there's trypsin, which helps digest your food, antibodies fight off diseases, insulin is a hormone. You see, they really do everything. Okay, let's move on to our next superstar, the polysaccharides, or what you might know as carbohydrates. Now, think of these as the cell's money. It's energy currency. Just like you need rupees to buy things, your cells need these carbohydrates to get any work done. So how does the cell store all this energy currency? Well, just like you put your rupees in a bank. These polysaccharides are just long chains of sugars. In plants, their energy bank is called starch. In animals, like you and me, it's called glycogen. And we store it right in our liver and muscles, so it's ready for when we need a quick burst of energy, for running or for studying for that exam. Next up, we have the boss of the cell, the nucleic acids. We're talking about DNA and RNA. Now, these guys are not the workers. Oh no, they are the master blueprint, the secret recipe book that has all the instructions for everything the cell ever needs to do.
And just like proteins are long chains of amino acids, these nucleic acids are long chains, polymers, of something called nucleotides. They carry the genetic recipe from our parents to us, and they're the ones giving out all the orders, telling those protein workers exactly what to build and when to do it.
And finally, our fourth superstar, lipids. You know all those food reels on Instagram? The ones with the sizzling oil in a pan, or those amazing cheese pulls on a pizza? Yep, that's all lipids. They're the fats and oils that we all know and, let's be honest, love.
But wait a minute. It's time for a trick question. And this is a favorite in exams, so listen up very carefully. We said that macromolecules are big, right? Over 1,000 Daltons. But lipids, they're actually pretty small, under 800 Daltons. So why on earth did they end up in the big stuff fraction? Why weren't they in that liquidy, soluble pool? Take a second and really think about that one.
Got it? Well, the answer is actually pretty simple. It's because lipids make up our cell membranes. So when we grind the tissue, the membrane breaks into all these little pieces. And these pieces don't dissolve in water, so they get caught in the filter along with the other big guys. So please remember this. Lipids are not strictly macromolecules.
All right, now for a very, very special topic. Enzymes. If proteins are the workers, then I want you to think of enzymes as the super efficient managers who make all the work happen at lightning speed. Honestly, without enzymes, life would be just too slow to even exist.
Just how fast are we talking? Let me give you an example that will blow your mind. There's an enzyme called carbonic anhydrase, and it speeds up its reaction by, wait for it, 10 million times. That is 1 crore times faster. Imagine something that would normally take you an entire year to finish. This enzyme gets it done in about 3 seconds. Wow, that is the power of enzymes. So how do they pull off this magic trick? This graph right here shows the secret. You see, for any reaction to get started, it needs a little push of energy to climb over this big energy hill. We call that push the activation energy. What enzymes do is they act like genius engineers. Instead of making the reaction climb all the way over that huge hill, they basically build a shortcut, like a tunnel right through it. By lowering the activation energy, they make it super easy for the reaction to get to the other side.
All right, my brilliant students, you've learned so much already. Now it's time for a quick-fire revision round to make sure all these facts are locked in for your exam. Are you ready? Chalo!
Okay, here's your question. Now, I told you earlier that collagen is the most abundant protein in the animal world. But what is the most abundant protein in the entire biosphere, on the whole planet?
And the answer is RuBisCO. Yes, it's that super important enzyme in plants that captures carbon dioxide for photosynthesis. Now listen to me, Butchin. If you remember just one thing from this whole explainer, please let it be this name, RuBisCO. I practically guarantee you will see this question in your board exams or NEAT. Mark it with a big, giant star in your notes right now.
And just like that, we're done. See, that wasn't so hard, was it? Just remember, you are literally made of all these amazing, hard-working molecules, and that means you, too, are capable of doing amazing things. So now, go and absolutely ace that exam. All the best!